Hi, Sujata. Uh, okay, so your question is, will you please explain the concept of CRISPR and how can we how can we make use it for editing? Okay, well, uh, I think you have first attended that course. How can we make you must have attended this course, right? And the lectures you have listened where we have discussed in detail about the CRISPR. But in short, we are like CRISPR, as it pronounced as clustered regularly interspersed short palindromic repeats, and this is the hallmark of bacterial defense system that forms the basis for CRISPR-Cas9 genome editing technology, right?
So in that, what happens is when foreign DNA comes in actually it's a bacterial mechanism in which when foreign DNA comes into the cells, there's a target sequence which bacteria acquires in between the Cre sequences, right? And after that, it form the transcription, and that means the bacterial genome had acquired the target genome, and after that, Cre RNA biogenesis happen where the Cre RNA binds to it and form the complex with the Cas9 protein, and then since it contains the complementary of the target sequence, so it binds to the complementary sequence of the foreign DNA and cleaves it, as it is cleared by Sir's lecture also, where they had discussed about it. So if you have any specific doubt about it, you can point it out. We can explain you that in detail.
And in fact, it can be used to target and modify typos if it's there in human genome. Exactly. So specifically we can target that and treat the genetic disease. That's what we are trying to research.
So this CRISPR-Cas9 makes a cut, and when the end joining happens, it always errored end joining because of that mutation in particular that gene happens, and that gene become inactive, or it can active also if required.
Yeah. Good evening, Gautam. Yes, Gautam, if you have any question, or Sujata, probably if you are not satisfied with the answer, or if you want more clarification or something, you can just write to us here and we'll try to clarify that doubt. And you can also share about, about the course, that how did you find the course, lectures, assignments, because till now all the assignments has been released.
Exactly. The course content is also released.
Whether you are finding difficult, or it's easy to follow, or you want something to include it, or any kind of inputs, we'll appreciate it.
If you have some difficulty in assignment questions or something like that, you can ask. Good evening, Nabi.
Yeah, if you have any query, we'll be happy to answer your queries, right?
Can you please give your ideas in restriction digestion? So restriction digestion, what happens is restriction enzyme or the enzyme which cleaves the DNA at the specific sequence. So we use them in the cloning. But what exactly you want to know about the restriction?
You must have heard that for and restriction endonucleases, we say that it's a molecular scissors. So that means to cut the sequence, we use it. Exactly. So it's a very clear thing. So in fact, it's now there are two kind of after cleavage also two kind of
end generates, the sticky end and the blunt end,
right?
And actually it really helps us for the cloning of the gene in particular, like vector or something like that. So if you have any specific question about the digestion, you can ask.
And lab session was excellent. Lab session could be arranged in nearest IIT so as to understand it better. Yeah, that I think almost all IITs they generally conduct that internship kind of program, or yeah, that is always available. And if any of the things even in this hangout thing you want to see in the lab,
we can show you.
We can show you with this with the help of probably this in this video also we can show you if something you want to see.
Otherwise, everything was in the video. We already had explained the instrumentation we are using.
In fact, if you want to see, we can show it to you in the lab because we are conducting. That's why we are conducting this
session in the lab.
So that if something specifically you want to see or understand, we could give the demo kind of thing.
Yes.
Right?
Yes.
I think that would be helpful.
We can give you the demo if any technique specifically you wanted to know.
And for restriction digestion, Gautam, you have to think about specific, like what kind of enzymes you are using, what kind, where do you want to cut? If you are doing cloning, then you have to see that what part exactly you want to put for your like whole gene, a whole coding portion you want to keep, or a specific some domain you want to keep. Accordingly, we decide the restriction sites and then we digest it.
So Sujata want to know if we can demo on any of the technique explained in the theory. So in the theory, we had explained microarray DNA sequencing. Those are very sophisticated techniques.
But PCR or gel running techniques, if you want, then we can show it to you.
After at the end of this hangout session,
or in between, we can show it to you anyways. Like if you want to see PCR or like gel egg-rose gel electrophoresis or Western, then we can show it to you Western apparatus. Even for RT-PCR, we can show you it to you the instrument and what kind of strips we use it, how do we decide it. We can explain, I think, even Saloni, this for RT-PCR also.
We can show you the
we can show you. So you just point out any of the technique, the one we have listed. We'll be happy to show it to you. Okay. Okay, the next question is from Ananya. The next gen sequencing method was a bit confusing. So, okay. So next gen sequencing, I think then probably you would have understood before this that Sanger sequencing and then before.
So in next gen sequencing is done by synthesis only. Once there's a sequence which is like they're attached onto the platform by the means of some small probe, and once they're attached to that probe, and after that the synthesis happens, and during that synthesis process the nucleotides were either the tagged or the pyro is the principle used in pyrosequencing also. Both those kind of principle can be used to monitor which nucleotide is incorporating. In that way you can like get an idea which nucleotide is coming next. So that is the way to sequence it. So there is one beautiful video sir also shared. In that the person has explained it in very detailed manner. I think
in the reading material link we have also provided that one.
So I think I also find it like more understanding, and my understanding also on this topic developed by looking at that topic only, that video only. So you can go through that video for one more time. I think things will be clear. As in there's a platform where the probes are, and then you put your sequence, and those probes have the complementary sequence with the DNA, and then the synthesis happens.
Basic principle is more or less similar to that Sanger sequencing.
By synthesis.
Sanger sequencing or any of the we have discussed. So which relies on the capillary electrophoresis, and that for initially you have to fragment the genomic strand, because until you fragment it, you cannot get to know that what's there.
It happens like this. It happened in the pyrosequencing, also sequencing by synthesis, but it's more sophisticated way to sequence and more. I mean, error-free and a high throughput technique.
And signals you can read out by emitted signals when the fragments are ligated against a template stem, because it will give the.
So this is how you do it.
So okay, Sujata, RT-PCR demo for RT-PCR. Okay, RT-PCR. Okay, I think we can take the lab.
The machine.
We can take the lab.
Let's have a tour in our lab.
Yes, I think that will be helpful. So we'll just take a. So Salome, I think you can take the RT-PCR strip as well. So then we can.
So
I think keeping laptop somewhere here. Here it will be helpful, right? No, you want to use that machine. Huh. You come here, I'll just do the setting. I think from here it's visible. Whole thing, right?
So
Software also we can show it
exactly.
to them.
So this is the BioRate RT real-time PCR system. So what we do is we first make the
Turn. On
so,
करना. This is a strip normally. It's
so these are the RT-PCR strips. Actually, for the RT-PCR, we use different kind of strips. So in that, as you can see, there's small amount of samples. So this is the sample. For RT-PCR, you use cybergreen. In this, I'm using cybergreen. So we put, as the reaction mixture, we take cybergreen, primer, and the cDNA, right? And after that, after preparing a mixture, after preparing the sample, you come to your machine, switch on the machine, and this machine is completely linked with the software. You need not to do with anything with the machine. All you do handling with the software. So
I mean, anyways, the machine can be directly operated, but it's always good to use with the software.
So software will take some time to detect the machine. Maybe
by the time you can already saved some program also.
Exactly. So I can tell you something about it. For example, in normal PCR, you put the enzyme, the buffer, and the NTPs, and the primer, and the cDNA, everything you put in the resin mixture. But over here, in the cybergreen mix, you need not to put the enzymes and the DNTP separately. All you use cDNA and the primer and the real-time mixture as the mixture provided by the BioRate only. So,
and for primers, either you use primer, and two of the things we use, either cybergreen or Tacman probes. Tacman you have to already that will be attached in this fluorescent probe in the primer itself, and cybergreen it will go and bind to this.
Exactly. And principal sir explained it in very
so here we can show you the demo. So probably now
once the machine is on, so it always comes there option. So you can first open lid so that machine will open. So it is all automatic. And you put your tubes in the small holes. These are the PCR. To put your PCR strip, you put your PCR strip like this. Once you put the PCR strips, then you go for the program setting, where you first choose the run type, which I'm choosing the user defined. And then actually this is the program. So I am getting I'm actually selecting my previous program for this. So what happens is, in this cycle, the first is a denaturation step, which is similar to PCR, and then there is a denaturation step for each cycle, and then there is this annealing step, and I'm repeating this cycle for the 40 times, as in for the amplification of the cDNA. And for the real-time PCR, the melting curve is also important to know if the primer is specifically amplifying the gene of interest or it's non-specific amplification. So once you put this program, then you go for plate editing. So in plate editing, what you do is you put your sample. Actually,
you to know that where the sample is. Otherwise, after reading, how will you get to know that in which well what you have loaded? To get to know that you have to have labeled the wells, right?
So I had put the samples in these lanes. So I will name those lanes so that I'll not forget what my sample is. So for example, if I'm using the primer for some gene HSP70, so I'll put the name of the gene like this, and if it is treated, it's control or the treated sample, so I'll put that sample name as well, control, and this one is treated, right? And once you're done with the editing, you go to the start run cycle and you close the lid. So it's all automatic. Lid is closed by itself only. Once it will be properly closed, then you can start the run. So
then only this option will be highlighted. So it's nothing is difficult. You have to just observe and follow the protocol.
So you will start the run of the cycle. You will save your program for the future purposes, and the cycle will start. So it's taking a time. Actually, machine is taking time. So now it's starting initialized. That is, it is starting with this step, the first initialization step, and then it will repeat it every time. And this program will take around Rashmi 1.5 hours.
So we can just directly go to the result that already you got, and you can just explain it that how we take control and I mean what exactly the control means. And
so at the end of your experiment, you will get the results.
This you have stopped it.
It's running. Program is running. So the result will come like in the Excel sheet. Obviously, the final data is in the form of Excel sheet, where you can get. So these are the target genes which I had used for my experiment, and these is actually I was putting the name. I think you must have observed. This is the sample type if it is not treated or treated, and these are the CQ value. CQ value is actually the value at which the amplification starts. So, for example, if your cDNA is too much, then amplification will start at the earlier cycle. That means CQ value will be lesser. If the CQ value will be lesser, that means there is the abundance of that particular gene in that particular sample. So on that basis, you do the calculation and find out your result that which gene is expressing more or not. I hope this demo helps you.
I think we are able to show you the RT-PCR thing. And now if you want to ask something more on this, we'll be happy to clarify. Probes with Jasmine. Okay. Thanks, Sujata. You liked it. And Jasmine, which probes? Probably she's talking about the sequencing probes
Sequencing probe, I guess.
I guess
those are the complementary sequences which are present in at the end of the DNA, with which it should be attached. I think Sir had covered
that in fact
so these are actually more or less like the primer sequences, which you use for the amplification also. So primer sequences are something which you know about some gene. So it's like that only 8 or 10 nucleotide base.
It's a kind of then because it will be 8 or 10 right? And it's labeled with the fluorescent dye. So then it that's why it cluster makes the library
cluster kind of thing.
Probes and which have a different fluorescent dye at the 5' end and hydroxyl group at the 3' end. And bases are complementary to the nucleotides, which has to be sequenced while base like 3 to 5 are degenerate. But and bases 6 to 8 are inosine bases. And only a complementary probe will hybridize to the target sequence.
Exactly. Non-targeted will be washed out, and the ones with the probe which is hybridized, they will stick on that platform, and that will be further sequenced.
Because then DNA ligase will use to join the 8-meric probe to the primer, and then it's used. Okay. Do you have the blast tool to show us? Blast tool actually tutorial. Blast tool we do. I mean it's available in the online. So while doing in this, it will be
showing you very difficult.
Or in another laptop, we can show it to you. Yeah, that's another option. We can show it to you, and I think blast. Okay, we can show it. That's not an issue. And for details, you can go by this tutorial in week 3 lecture. No, week 4. Week
blast 4 lecture in tutorial session 1.
Week 3 probably. Blast 1 week 3.
Week 3, I guess I had covered sequencing and the microarray thing. In week 4, I had covered the evolution thing.
Anyways, 3 and 4. Blast tool, blast, and everything was there. So there also you can see. Otherwise, we have bit of we can explain it from the another laptop. That's not an issue. Okay. Let me see.
I guess it will be a mirror image, Rashmi. I don't think it will work. So, okay. So, okay. So what happens is you go with you search for the NCBI blast. So NCBI blast, you'll go. There are the different kind of blast: nucleotide blast, protein blast, blast X, and the primer blast. I'm choosing the website actually. So this will be the home page of it. So where it is showing all the alignment tool, where the blast X is nucleotide to protein, T blast N is for protein to nucleotides, protein blast is for protein to protein, and nucleotide blast is for nucleotide to nucleotide. You'll go according to your preferences. So I am
according to your query, you have to take it.
Ki what kind of sequences you want to align. So I'm choosing the nucleotide alignment. So you there are the
either here you can put the accession number directly, or if you have a sequence,
FASTA sequence also.
Any of the so you can just give a quick search of any of the gene sequence.
So gene sequences. Okay.
In another tab.
So what you will do is
NCBI again.
So you'll go with the gene sequences.
Nucleotide directly. For
I'm going for the
EPM2A or HSP70 anything.
HSP70 I'm going with. So you search for nucleotide HSP70 and
you can take your interest of gene
and species, host species, what you're choosing.
You have to see.
I am choosing for the HSP70 FASTA sequences, and there is some Toxoplasma gondii HSP70. I'm choosing its FASTA sequences.
It's just for reference. Any of the randomly we are taking.
Taking. So this is the FASTA sequence. So what you can do is you can take the small part of it. I'm not taking the whole gene.
So that it will be faster. That's right.
So I'm taking this small part, and then I'm going to the tool, putting it. One.
I think a whole genome set also you can
So I'm putting space. So align 2 or more sequences. So I'm.
Or directly you can just with the database also you can go.
So okay. So So and the other one is this. So I'm taking this FASTA sequence as well. I'm taking small portion only. So it's for just the demo purposes. So you will put it here, right? And after that, you will I'll show the result in the next window and blast. That's it. I'm going with the default setting. I'm not making any difference in the program.
I mean, you can read.
So I guess in Drosophila and the text took the species which we selected, the Toxoplasma gondii, the sequences in which there is no significant similarity. That's why the window can say no significant similarity is found. So this is how you do the blast.
So if there are similarities, then you can easily see that was similar sequences. I mean, same type of the one we have shared in the lectures.
So,
question 10, assignment of week 3. What was question 10?
I think we need to go for the assignments.
We'll just see that. Let us check that. What was the question 3? Week 3. Okay. Researchers often use the basic local alignment search tool to find the homology or regions of similarity between amino acid and or DNA sequences, which one of the following statement is incorrect with regard to this approach. Okay. Identify intron-exon boundaries. Yes, this you can do it because we have asked that what cannot be done with the help of BLAST tool. Okay. So anyways, you can identify intron-exon boundaries with the help of BLAST tool. Identify potential coding region of the genome. Yes, nucleotide blast you do, and identify presence of transposable element in the genome. That also you can do it by the blast tool. Then there is none of these. None of these means any of the option is not can be done by the blast tool, right? So sorry, none of these means all the above three are incorrect, but that's not the case.
In the blast tool, I can find out these three things.
So,
all these three things.
That's why the answer is none of these. I think Sanmati, now you would have understood that what we were saying. Can you please also suggest some reading material or sites apart from books for reference?
You can always go through the Google, as in if you have any topic.
Okay, we'll give you, although whatever, if for exam purpose you are asking for this particular course, then whatever the lectures are there and whatever we have shared reading material, that is enough. Otherwise, anyways, we'll provide you some reading material link. That's sites as well, and that's not an issue. I mean, even that is sufficient. But if you are interested for something specifically, some technique or some specific topic, then we'll be happy to share you some recent research article. I think that will be very helpful for you. Although if you go by previous, previous answers, you will find that we have shared links for the reading material apart from the books as well. That what can be the used as advanced genomic approaches or something. Abhay, please suggest any reference book for the course. Okay, Abhay, reference book we have already shared. That for very basic things, you can always go by this biology by Campbell and genetics by Griffith and cell and what was that? T.A. Brown for genome and Lordish. All these are very basic one. And for the advanced new technologies, I would rather prefer to go by recent research articles. That will be very helpful because they'll tell you what the latest technologies we are using, what the techniques we are using nowadays. That will be very helpful for you. And we have already shared for this reference books and all the reference material. And in fact, new technologies link details also we have shared before this as well. Okay, so next question is from Mahima. What is morpholinos? So morpholino is again a oligomer and as a phosphodiamidate morpholino oligomer. So normally it is PMO and it's a type of a oligomer molecule which is used in molecular biology normally to modify the gene expression, right? And the molecular structure of it has the DNA bases attached to a backbone of this specific methylene morpholin rings. That's why it named as for something rings and linked through this phosphodiamidate groups. And morpholinos normally it blocks access of other molecules to small specific sequences of the base pairing surface of RNA. And it is used as a tool for reverse genetics by knocking down the gene function. Okay, so next question is by Sujata that what is this? Metagenomics and its importance. Okay, so wait. So metagenomics, it's again a high throughput DNA sequence analysis and it's an important tool for this. because we use for immense richness of the diversity of particular community, which present a strong mandate for the use of functional metagenomics strategies that involve broad screening methodology to discover the study, right? In fact, in that we have given one example where it was used that if they were staying in um, particular habitat, and when they move, and then how it can be correlated. So that's how metagenomics is used. I mean, it's a very dramatic effect in the way we view the study of microbial world as well, in the case of metagenomics. Any other question? For importance also probably she has asked. Okay, importance actually it has applications of metagenomics in various like bioremediation, industrial byproducts, and application in the human microbiome. Like for bioremediation, it's used widely. Industrial byproducts also it has been used because like for the mining the rich genetic resource of the non-culturable microbes has led to the discovery of new genes, enzymes, and natural products. And the impact of metagenomics, it's actually developed the commodity and the fine chemicals, agrochemicals, and pharmaceuticals, where the benefit of enzyme catalyzed chiral synthesis is increasingly recognized. And in the human microbiome, in plant microbiome interaction, it has wide use. Any other question? No more questions are coming.
I think they'll ask. We should wait.
Okay.
How can we do whole genome?
Okay. How can we do whole genome comparative study? Shall we? Yes, entire genome and the annotation of this. Because when you are going for whole genome study, then you have to see like whole genome comparative for with one organism to other, what you want to see, that you have to check it, right? So for to adopt this, like for this whole genome comparative analysis, there are techniques. Comparative genomics, for this is the main thing that we use. It can be used for anything like plant pathogen or anything. So you have to take all the context and everything. And that's the same. Like we said, the different organism you have to compare. You have to decide that which organism you want to compare with with which one.
And then you take the advantage of next gen sequencing and that quantic approach where you can like they're the overlapping sequence with which you construct the whole sequence and then they compare it between the species. And yes, it then helps. This is how it helps in the comparative genomics.
Because it will with the genomic feature, it will include everything: DNA sequence, genes, gene sequence, everything. Week 1 assignment question 2. I think the weather can identify genes with shared function. Can you explain why is it wrong? Okay, if I remember correctly, it was the question that only the reverse genetics can. In this kind of way, I have to just go through again. But if I remember correctly, because that can be done by this as well. Forward genetics as well. Which question she was asking?
Week 1, question 2.
Question 2, right?
So she's asking, research understand the function of the gene.
Either the forward genetic approach or reverse genetic approach. And which one of the following statement is true? Only for the, that's what I'm saying, that only for the reverse genetics. That cannot be done by forward. So shared function, anyways, forward genetics will also tell. So if you go by this, understanding gene function at organismal level, that is also can be done by both. And can identify genes with shared function, that can be done by both. But for redundant function, we cannot determine by this forward genetic approach. That's why option, this redundant function is true, because we are asking only, you have to just read carefully, that true only for the reverse genetics approach. And with the solutions also, that's why we have provided the explanation on the same. No, I mean, Samati, you were right that reverse genetics can identify genes with shared function. And it can be done by forward genetics as well approach. So that's why it's not right answer, because that has to be specific for reverse genetics, and that is the redundant function. Okay, nice. Okay. Okay, can you discuss a bit about what you work in the lab? I know the lab works on Lafora disease. Oh, really interesting.
Which lab, you know?
And but just curious to know, what is that the lab focus on? Okay. So our lab, basically, now you know that Lafora disease. So Lafora disease, it's a neurodegenerative disorder, and it's a type of progressive myoclonic epilepsy. When we say progressive, that means with age, it like whatever the symptoms, they are getting severe. Okay. So normally, if you want, would you like to get into more detail of Lafora? But I'll just give you the quick information about our lab. So for this, we know that two genes are involved in this, and there can be third or more, because there are patients where we do not find the mutations in these two genes, but still the Lafora symptoms were there. So we think that there can be another loci as well. And for this, we use cell culture and animal model to identify the particular pathways, what are the molecular pathways involved. And now we are more of in the towards the therapeutic approaches, that how can we treat this disease. So we are targeting that particular pathways, markers, and then we are trying to see that particular drug is working. Like, for example, I can just share, if you are familiar with the autophagy term, then we can say that, okay, autophagy is impaired. Then we can think of that, how can we restore that particular pathway without its any harmful effect. Okay, because while restoring that particular pathway, you have to keep in mind that you are not, you have to see that it's not harming the animal in another way, that for restoring one pathway, but it has some negative impact on another pathway, then it will not solve our purpose. So we have to keep in mind all this thing, and then we see that whether these are the things working or not. So first we go by the cell culture models, and then we go and check on animals. That's how we work. And our lab is not limited to Lafora disease, but yes, majorly work on Lafora. Other neurodegenerative disorders also we target, like Alzheimer's, Huntington's. We do work on that. And myopathy thing, and like some heat, stress response, but probably in this, no, that was in professional scientific communication, that our lab work was discussed. Okay, that was some different interaction thing. So apart from this, also we work on cellular stress pathways, that how stress,
how cell responds to the stress conditions.
Right.
And also in Laffer disease, also we explore what are the pathways majorly which are impaired, so that we can target them for the therapeutic purposes. In nutshell, I guess.
Yeah.
So you can go through the papers also from our website, from Sir's website.
If you go by lab web page, you will get in detail.
In detail, what kind of work we are doing.
But in short, that's we work on this. I mean, we are trying to
find
decipher,
the solution.
Find the solution, that trying to decipher that already, although we know that several molecular pathways are involved, and
we are trying to target them.
Target them. We are that what are the molecular markers? How can we restore them? And I hope, Ananya, I mean, if you want to get into the detail of any of the specific things,
you just the specific question.
We'll be happy to answer if you want to get into more detail, because I have just given the overview so that you know that what we are working.
I think Rashmi.
Okay. Yes, almost, Sujata, we have discussed whatever the recent years is naked mole rat. It's a really interesting model nowadays. And Ananya, thank you. I'm really interested in the kind of work. I wish to write to sir sometime if I can work at his lab. Definitely, you are always welcome to write to sir, and we'll be also happy to see you if you are interested to work in our lab. That's really interesting. And for details, then if you are really interested in working in the lab, then I would rather suggest to go by the web page and see that what kind, because whatever the papers, web page is properly updated, so you can find the papers, what kind of the work we are doing, short description is also there. And always you are welcome to write sir, and one of the lab member. Anyone will be happy to answer your queries. And this naked mole rats, they are definitely, I mean, it's a very fascinating rodent model, and because of its weirdness, it's this discovery made the researchers made fascinated about this one. That how can the animals can survive up to 18 minutes in an environment without oxygen? That's really
that's the advantage of this model. It can survive without the oxygen up to 18 minutes.
Because
I think it can be used for studying the hypoxia, as in during the
because where then you can relate that how the pathways are working there. Then you can where the hypoxic condition is, then you can just utilize that particular thing, that how that particular animal model is responding. And
exactly. It can have several advantages of.
It has actually. So that will be actually will be helpful for because hypoxic condition, it's again a health health problem. So for treating such kind of health issues, it will be very helpful. Because they have sensory bristles on their skin, and they are too much sensitive. So how do they behave? How do they acquire all that senses? And although eyes are very small, but they can see clearly. I mean, they are not blind, and although sometimes but they have poor vision, but they can see, right? And they use eyes, because there was one research which says that they can use their eyes to distinguish light from dark, and there's no external ear flap, only hole is there.
So there are very interesting feature on that mice which can be used for the research purposes.
And in fact, you remember for this naked rat model, it has social organization as well, that resembles a bee colony kind of thing. That has also there. So that rodents and insects are very different animal, but still it has similar kind of features. So I mean, there are several interesting and surprising fact about this naked
Particular
mole
animal.
that. And this like this skin of this naked mole rat, it cannot detect pain caused because by acid or by capsaicin from hot peppers, and it could be very useful in tunnels filled with exhaled carbon dioxide, since the chemical reacts with water to form carbonic acid. And also a very interesting fact is that this animal don't get cancer. So it might be more accurate to say that they rarely get the disease, because saying this that they don't get cancer, it's a very, I mean, big thing. So it's better to say that it's they rarely get this disease because of at least two animals in the captivity have developed condition that resembles cancer, not exactly the cancer condition. Thank you, Sujata. It's really nice when you guys give compliments, right? And for us also, it's very nice to see that you are liking the course and appreciating
the session and everything.
Right, because it matters a lot when we are teaching and someone is getting
always makes
and someone is getting benefited. That's our motto. So it should be. And thank you, because you guys are also very particular. That really
exactly. You guys are interacting. That's why we are interacting. Otherwise, it's not possible.
It's always it's appreciable that when you see that students ask question and everyone is interacting, then we also feel happy that okay. I mean, you are following the lectures and assignments. We really feel happy whenever we see the questions that okay, you are
even a discussion forum also, when people ask questions, it seems like okay, they are reading something. So it makes us happy, and we are happy to answer. In fact,
thank you for coming on the live session.
I think no more questions are coming.
Let's wait for 5 minutes. If no one is coming, then we can end this session, right?
Exactly.
Okay, Saurabh. Yeah, has local chapter for NPTL. Do we have that internship modules for internship module for this course? I don't think so. But for internship, like if you what you're doing, masters or BTech or BSc, whatever, if you are interested in doing like a 6-month internship also, so then you can directly contact to sir or any of the interested faculty. You can contact them, and if they have vacancy for internship, they'll reply it to you. Okay, nice, Ananya. That
you attended the other course also.
And yes, and you like the course, both of the courses.
In fact, our laboratory name is also Human Molecular Genetics.
Our basically
web-page says so.
Because we worked on
molecular genetics.
We have worked on.
And we are happy that you learn from sir's courses. So.
Definitely, he is a great teacher. That we do, and we do say.
We also say that
because we have learned everything.
He always makes the complicated thing in the simple when then he explains. This is something which we really like.
And due to some unavoidable
circumstances, he is not available.
He could not attend this live session. Otherwise, he would have been here.
Exactly.
but anytime you can write him, and
you can contact him.
So his mail ID, everything is available. You can write to him, and he'll be happy to answer you. Okay, Sujata. Probably either this, at least till now, whatever we from our lab, we have 3 courses were enrolled. One is functional genetics, genomics, and another was human molecular genetics. And apart from that, we had introduction to professional scientific communication. Till now, these 3 courses we did it. And in future, probably we'll be rerunning these courses, or we might come up with some new courses also, because if you could if you have any specific interest, you guys always give your suggestions and feedback, so we'll think accordingly. Also, we can plan that. We can plan the course as well. Okay. Be in biotechnology engineering in May. Okay, so after that, if you like for a master's also, if you have something, thesis internship kind of thing, you can always apply, and you can always mail and see whether the vacancy is there, and then you can apply.
You can apply for the project purposes. You can apply when you completed your BE. You can apply for MTech and PhDs also.
You write for GATE and all, and if you clear the exam, then you can come here for MTech program or PhD program, because after BE also directly BE is like BTech, right? Yes. So directly you can apply. Degree, I guess. So I think it's the same. You can go for a net CSIR, GRF,
any fellowship,
any fellowship, and GATE. So any of the fellowship, if you have, then
you can apply over
JRF, and with this GATE thing, you can apply for this MTech and PhD program here.
And then there's a written exam also. You qualify that, and you are in.
Written and then interviews. No more questions are coming. I think let's see for 2-3 minutes.
His time is also it's running. So it was really nice to interact with you guys.
Hi. And after the session, also you can always email us your feedback, and
you can always ask the question or discussion forum, and we will always be available to answer that.
And we'll appreciate your feedback as well. Yes. So.
I think which means the time is.
Time is also now. So I think if there are no more questions, then we can just sign out.
I guess so. It's time to say.
Just let's see for 1-2 minutes, because probably there's some lag.
Okay.
Okay, participants, I think we are not getting any of the queries now. And thank you for whoever has joined us. And those who could not join us, it's they are always welcome to write and ask the questions in the discussion forum or via email. Everything will be appreciated. And okay, so now we are signing off.
Yes.
I think. Okay, thanks.
Thanks a lot for joining us.